So I would just like to welcome everybody to the United Leukodystrophy Foundation's virtual family conference session on ALSP HDLS, presented by Dr. Eichler, Dr. Orthman Murphy, and Heidi Edwards. This session is being recorded and will be posted for viewing online in early July. Thank you so much for joining. And now I will turn it over to Heidi Edwards' session.
Heidi Edwards, president and founder of Sisters Hope Foundation, a nonprofit organization supporting families affected by adult onset leukoencephalopathy with axonospheroids and pigmented glia, or ALSP. Some of your family may have also been diagnosed with HDLS, hereditary diffuse leukoencephalopathy, or POLD. My journey with ALSP started over 20 years ago when my Aunt Ruth was diagnosed with what they thought was a frontotemporal dementia, called Pick's disease. She passed away in 2005, and her brain tissue was donated to research at the University of Pennsylvania. Her autopsy showed a diagnosis of Binswanger's type dementia. And at that time, we thought our family did not have to worry about a hereditary disease. The experts felt it was just one person in our family and no one else had anything to worry about. Until 2009, when my mother became symptomatic. She also started showing similar symptoms to her sister. She again went to the University of Pennsylvania. And at that time, they told us that we were looking at a hereditary condition, but they truly were not sure what it was. She was clinically diagnosed with cortical basal degeneration. My mother passed away in January of 2012, and her autopsy revealed she had agorophilic brain disease, something different than her sister. Within the same time frame, my uncle was symptomatic, clinically diagnosed with Lewy body dementia. He passed away in 2013. And at that time, the Mayo Clinic in Jacksonville had found the mutation, the CSF1R gene. So when my uncle passed away, our family was tested through their brain tissue, and it was determined that they all had the same mutation, the CSF1R mutation. Our family was diagnosed with hereditary diffuse leukoencephalopathy, or today it's known as ALSP. We thought we had some time to figure out what was happening to our family, but quickly after the three relatives passed away, my two sisters became symptomatic. Heather and Holly were symptomatic for the disease by 2019. In their early 40s, they were showing the same signs and symptoms that the other family members presented with. My other family members passed away in their 50s. Heather and Holly were in their 40s, and we really weren't sure what was happening. Why were they 10 years younger and presenting with symptoms? Heather and Holly both were tested for the mutation, and it came back positive. Heather decided to undergo an experimental bone marrow transplant at the University of Minnesota. Heather started her bone marrow transplant, and I was her donor. She started the 10 days of very intense chemotherapy. She did suffer a heart attack after 15 days from the date of transplant. Heather never fully recovered. And on August 28th of 2020, she passed away. In the meantime, Holly decided she did not want to undergo the bone marrow transplant. She did start the process of all the testing, and she did have a donor ready to donate. But after witnessing what Heather went through, she decided it just wasn't right for her. It didn't feel right. And she wanted to come home and live out the rest of her time surrounded by her family and friends, and especially her son, Mason. Holly, all she wanted to do was see Mason graduate. And last week, he graduated from high school. We are now spending our last days with Holly, her last days with her. and enjoying every moment we have with her. I am determined to help others who are affected by ALSP. I have watched 5 family members struggle with this disease. From time of diagnosis to the time of death is about 2 years. It's extremely rapidly progressing. And at this point, the only option is a bone marrow transplant. And researchers and scientists have now found that you do have to have the transplant done early before you have too many symptoms of the disease. My family hasn't been fortunate so far. 3 passed away in their 50s. Heather passed away from the bone marrow transplant, and Holly decided to live out the rest of her days surrounded by her family and friends. And that's what she's doing, living the best life she can. But we still have no cure for the disease, and we're going to lose one more family member. I started the Sisters Hope Foundation to honor my sister's mom, aunt, and uncle. For so long, my family and I felt alone with this disease. Through Sisters Hope Foundation, we are raising awareness, education, building a support network, and a community to connect families who are affected by this devastating disease. We provide resources to families, including information about genetic counseling and support groups, educational resources, and tools for patients and families. Sisters Hope will serve as a central resource for patients to learn about the newest information. We are working with neurologists, researchers, and industry partners to further research to not only learn about ALSP, but to eventually find a cure for this disease. Heather, Holly, and I have always hoped for a day without ALSP, so no individual or family has to experience the devastation of this disease like we have. The legacy of my sisters will last forever. Through Sisters Hope Foundation, help me honor my sisters and work toward a future where those diagnosed with ALSP survive and have a meaningful future. Thank you.
Thank you, Heidi. That was very moving, and a big reminder of why we're here together, and why it's so necessary to bring everyone who knows about this disease and can contribute something together to help better the lives of the next generation. Together with Dr. Jennifer Orton-Murphy, I will present an overview of ALSP, HDLS, or CSF1R related leukodystrophy, and I will start off by addressing some of the things that Heidi spoke about, which is the difficulty around different kinds of names we're using for this disorder. And I want to just point out that while we're talking about the same thing, the names have changed over the decades as we understand the pathology and the genetics better. Originally, this was known as pigmentary orthochromatic leukodystrophy, or POLD. This originated from the fact that when in the 1930s, the autopsies were looked at, there were certain cells, microglia, that appeared pigmented. And a few decades later, autopsies in other adults with this disorder were then named hereditary diffuse leukoencephalopathy with spheroids. That was because on autopsy, there are certain swellings of the axons that were recognized. It was only 20 years ago that certain connections between these two entities were made. And it was pathologist at the time went back and was able to look at some of the earlier autopsies and recognized that both axonal spheroids and pigmented glia were seen on all those autopsies, and therefore the name adult onset leukodystrophy with neuroaxonal spheroids and pigmented glia was used. As Heidi mentioned, genetics has made advances, and we now have certain genetic markers, specifically CSF1R, that has now become the most common mutation for this leukodystrophy. And we're using the term CSF1R related leukoencephalopathy. There's some controversy over whether to use the term leukoencephalopathy or leukodystrophy. I tend to use the word leukodystrophy because it's really a progressive disease. And leukoencephalopathy tends to say that it's something more static, something that's not changing or degenerating. Leuko meaning white and dystrophy meaning degenerative. And so in this talk, I will use the term CSF1R leukodystrophy. So back before we knew about the mutations, we had first patients at Massachusetts General Hospital that presented with this disorder at the time still called Hdls or Alsp. But the pattern was very similar in all of these patients. They had predominantly a frontal white matter disorder, as you can see here on MRI, and enlargement of the ventricles. Note how confluent these lesions are. And if you looked at a certain technique called MR spectroscopy, and that MR spectroscopy, you can measure certain chemicals in the brain. There was a loss of NAA, N-acetyl aspartate, which is a marker of neurons and axonal health, indicating here that axons and neurons were affected. And on autopsy, what you often saw was this, this pattern here of axonal spheroids and axonal swelling. Others have described this pathology in greater detail. Let me just point out a few things that you see in autopsy. Everything that's white matter is stained blue here with Lux-Alphas blue. See how this is a confluent lesion that generally spares the areas right underneath the cortex, these subcortical areas. And when you use a certain staining called amyloid precursor protein, you can stain these axonal spheroids, and you can see them here on a simple H&E stain quite clearly. These kinds of balloons of axonal spheroids that have given the disorder its name. There are also a lot of these macrophages that look very activated and are found throughout the brain. And you'll see later on that this is an important finding, as really this is a disorder of microglia and how they're affecting these axons. Here again, some examples of the axonal spheroids seen here. And you also see a lot of calcification of vessels. So, around the vessels in the brain, you see a lot of mineralization. For providers and for families, it's important to keep in mind that there are certain patterns on MRI that you want to look for, and where you want to think of HDLS, ALSP, or CSF1R related leukodystrophy. And I always teach my residents that it's really the frontal predominant pattern when you see an MRI where there's a strong frontal lesion, that is where you want to think about this disorder and send off for genetic mutations in CSF1R. Beyond that, you can also see certain parts of the cortical spinal tract involved. You see large ventricles that have risen over time. You also see some calcifications that occur in these patients that can even be seen on a CT scan. So this disorder is found really throughout the world. This is not something that is limited to one particular ethnic group, as far as we know. It's been reported throughout the US, large parts of Europe, and as well as China, and many cases in in Japan as well. And I think it is vastly underdiagnosed, and we're probably seeing just the tip of the iceberg here. There are probably a vast number of patients who are not being recognized as having this disease and are misdiagnosed as either multiple sclerosis or frontotemporal dementia or a form of Parkinson's disease. But now that this mutation CSF1R is being added to gene panels throughout at least the US and Europe, we're seeing a dramatic rise in diagnosis that is occurring. And I will say, compared to just 2 years ago, I'm probably doubling the number of patients in my clinic just because these panels are now finding the mutation, despite doctors not necessarily knowing about this disease, but simply because the gene has been added to a panel on dementia or ataxia or Parkinson's. Just to point out that this disease is inherited in an autosomal dominant fashion. What does that mean? That means that you when you have one affected parent, they have simply one allele being affected is sufficient to cause disease. And if you pass that allele on to your child, that child will manifest the disease, as opposed to recessive disorders, where you usually require 2 copies of this mutation to manifest in recessive disorders. So the autosomal dominant nature poses some challenges. And you can also sometimes see de novo cases, meaning these alleles arising sporadically. And there are also many questions around full penetration of these mutations. In some families, it seems to be fully penetrant and all family members manifesting. In other families, we're not seeing that. And there's a lot that we still have to learn here. So what is colony stimulating factor 1 receptor? Colony stimulating factors are important for differentiation of cells from the bone marrow. These hematopoietic stem cells need to differentiate into various cell lineages, including macrophages, and this is where colony stimulating factor 1 is important and contributes here to the production of bone marrow. The CSF1, as well as interleukin 34, binds to the CSF1 receptor that is found on monocytes and cells of the myeloid lineage, and then it produces this homodimer that then, after binding, autophosphorylates and activates. So this is the important structure that we need to target if we ever want to get to a cure of this disease. And this is, I think, already big progress that we understand that, and that we have now a clear target in mind for future correction. So we've recognized that CSF1R being the disease-causing gene, and that gene and the expression of this protein being on microglia in the brain, these cells are the ones that are likely the big contributors of disease. What do microglia do? Microglia act a little bit like the traffic cops of the brain. They're the first responders to any injury in the brain. They help rush to the scene if there's infections or injuries, and if they're mutated or they're somehow harmed by mutations, they're not going to respond well to injuries. They're not going to respond well to the cleanup that is needed, or they'll give the wrong signals at the site of injury. So where do microglia come from? Originally, during fetal development, microglia arise from something called the yolk sac. But later during life, adult life, we can see regeneration of microglia directly from the bone marrow. And so thinking about this disease of CSF1R-related leukodystrophy or leukoencephalopathy, there are here possibilities that there's both harm during development, but also ongoing during lifetime, where we think that these faulty microglia are not helping sustain neurons that go through this ballooning process and spheroid formation, then leading to a more progressive demyelinating disorder. Most of the mutations that are found in CSF1R are found within a certain domain known as the tyrosine kinase domain. There have been a few mutations that are found outside of that domain that I know of that have occurred mostly in Chinese families. So what are the first symptoms that patients experience with CSF1R related leukodystrophy? And you can see here, this ranges from everything from cognitive impairment to gait disorders, dyskinesias, dysarthria, loss of weakness in one limb. But the predominant problems early on are really one of cognition and mood. And we often see patients who are suffering here from emotional ability and ability to multitask, often troubles with judgment, executive function. And this is a huge burden upon not only the patient themselves, but also upon families who are struggling on how to redirect patients, how to give them guidance, and also how to prepare for future events that might be related to poor judgment in those situations. So in our clinic, I've plotted below the current ages and ages of symptom onset that we've seen. Clearly, these numbers are rapidly changing as we're diagnosing more patients. I just want to point out that they all have their onset of symptoms after age 30, but as late as the 60s. Some of these patients are pre-symptomatic family members who have the same mutations as those that are affected. So there are certainly challenges here around understanding full penetration. So more than a decade ago, I met a family with presumed late onset metachromatic leukodystrophy. This was a 60-year-old man with progressive cognitive deterioration, motor problems, and he was one of 4 affected siblings. And the brother who was not affected came to me saying, we have to consider a transplantation here and also diagnose him appropriately because he has metachromatic leukodystrophy since he has a 35-year-old sister who had been diagnosed with MLD and received an allogeneic bone marrow transplantation. And this had happened 20 years earlier. And she, at the time, had stabilized in her disease. She was had some cognitive changes, was a little impulsive, but had, after receiving the transplant, done well and had normalized in her deterioration. So the disease did not, the bone marrow transplant did not reverse any symptoms, but no further progression occurred. What was curious is that there were several other family members and that were affected by this disease. And when I tested for the aryl sulfatase A in white blood cells, I found that the levels were normal, and in urine, the sulfatides were normal. So this was not metachromatic leukodystrophy. Well, what was it? It turned out to be disease due to CSF1R, and we performed whole exome sequencing and figured that out, and then tested the parents who I had not thought of an autosomal dominant disease at the time because the parents were both normal. Well, it turns out that the mother was actually mosaic, and that she displayed the mutation in some compartments, but not others, and testing blood and saliva showed that. And what was even more interesting is that the sister who had gone through a transplant was now chimeric, which meaning she was also now a mix of different cells that had the mutation and other cells that did not have the mutation. So this raised the question whether hematopoietic stem cell transplantation could have a therapeutic role for this disorder. And so in the subsequent years, both in Europe as well as in the US, several groups have performed bone marrow transplantations for this disorder. Here, a group in San Francisco and California, as well as in France. And essentially the idea of bone marrow transplantation is that you take cells here from the bone marrow of a donor, and as long as those donor cells are well matched, you transplant them into the patient. Those cells engraft in the bone marrow. They move from bone marrow to blood, and from blood to brain, and become healthy, normal microglia. I want to point out, however, that there is a significant morbidity and mortality from bone marrow transplantation. And this is a paper out in the field of leukemia that recently reported the rates of death after hematopoietic stem cell transplantation. And although the rates of death have decreased in the period comparing 1980 to 2001 to 2002 to 2015, still there's significant mortality associated with this transplantation. And hearing the story of Heidi's sisters and Heather and Holly and what Heather went through is a stark reminder of how dangerous bone marrow transplantations can be, and that there are many things that add to the risk of this procedure. So even after you've gone through a transplant and made it through the acute phase of the transplant and engrafted well and overcome graft versus host and other complications, there are other complications that can occur long-term that one has to consider. There can be cataracts, thyroid problems, lung damage, infections, also weakened bones, skin rashes. So this is not to be taken lightly. Even with recovery, there is chronic problems that one has to manage, and have a very good team to help through this. With that in mind, more recently, people have started to think about opportunities of using immune suppression to maybe be protective in CSF1R related leukodystrophy. And here's a woman in her 40s who was affected by CSF1R related leukodystrophy with a classic frontal lesion. But her mother in her 70s had a normal white matter, although she has enlarged ventricles. And the question was, did she possibly benefit from many, many years of an immune suppressive regimen that she was on due to a rheumatologic disorder that she had? I believe it was arthritis that was being treated at the time. It's very hard to judge from a single case, and we don't, again, know about the full penetrance of this mutation in this family, but it is intriguing that the combination of different immune suppressions might have contributed to a better course in the mother compared to the daughter. In the last few minutes, I'm just going to tell you a little bit about the Global Leukodystrophy Initiative Clinical Trials Network, short GLIA-CTN, where we are really working to help many leukodystrophies, such as Alsp or Hdls, towards clinical trial readiness. And I think there are many reasons to think that CSF1R related leukoencephalopathies or leukodystrophies are going to be a good target for treatment. What's the mission of GLIA-CTN? It's a consortium of different institutions and clinician scientists that are trying to raise awareness, provide advances in diagnosis and treatment. We're trying to standardize ways of assessing patients across different sites and centers, share data, share information, and work closely with a diverse group of different stakeholders to better the lives of patients affected by these leukodystrophies. Right now we have 8 sites, ranging from CHOP and Kennedy Krieger, MGH, Utah, Stanford, and centers in the southeast, southwest as well. And importantly, we have a wide collaborative network, and we're really looking to bring more centers on across the US and foster the talents of young investigators who might be interested in stepping into the field of leukodystrophies. So we collaborate with a lot of European centers, but are also looking to expand relationships and partnerships with other centers across the world. So this is led by Dr. Adeline Vanderver at CHOP, Dr. Ali Fatimi at Kennedy Krieger, and myself. And I wanted to point out that a big part of what we do is partnerships with patient advocacy groups such as the Sisters Hope Foundation, and really the patient's voice, the patient's experience and perspective is what drives progress and brings everybody together, recognizing that it is really that experience that we need to transform, need to change, and as Heidi said, bring about a meaningful difference for families moving forward. I think one of the projects that we are undertaking in the CLIA-CTN is a longitudinal natural history study, and that is a very good fit for disorders related to CSF1R, and understanding together how this disease progresses over time is going to be key to setting up first treatment trials for this disease. So I will end here and pass the baton to Jennifer Ortman-Murphy. Jennifer?
Sorry, I just had to take myself off mute. Thank you so much, Dr. Eichler and Heidi, for your wonderful talks and intros. And I'm really thankful to be included in today's session. So I can speak to you about what I have come to learn very quickly in the past couple of years about CSF1R related. I have here leukoencephalopathy. I'm also willing to use leukodystrophy. I like referring to the gene primarily at this point because that is what I can tell the person in front of me who I'm diagnosing and their families. So to start off, I kind of want to give you some perspective of where I'm coming from, because for a lot of the leukodystrophies, you don't necessarily come from the MS field, which is where I'm coming from. So it might help put into perspective some of the ideas that I will present. So the objectives for today is that I'm going to talk a little bit about the identification and care of people diagnosed with ALSP in an adult neurogenetics clinic, and as well as my ongoing clinical collaborations to better define this disorder and the disease course and develop a biomarker platform. And then I'm going to talk a little bit about the basics of the underlying disease mechanisms. You heard some of it already from Dr. Eichler, but I'm going to put it into context of my own basic science research program, which is on glial cell biology and fixing the brain. So, and sort of where I think we could go with it. So I'm going to use some terms a lot, and I just wanted to define them up front. So the CSF1R related disease is a white matter disorder of the brain. And so I just wanted to show you that we're often looking on brain MRIs, where this is a cross-section through the brain, where the nose will be up here on the top of the image and the back of the head is at the bottom, and this black filled space is the fluid. And the white area on this version of an MRI is the white matter, and the gray matter is this ribbon that's on the outside. And the reason white matter is white is because it's filled with fat, and the cells that make the fat are oligodendrocytes, and they form these highly complex structures called myelin sheaths. Myelin sheaths basically protect neurons, which send these long projections to other neurons for the brain to function. And in this complex network, we're thinking about not only these two cell types, but actually many other cell types within the white matter of the brain. We just don't have the resolution to see it on MRIs. But when we're thinking about white matter disorders, shown here is the version of the MRI that I'm often looking at as an adult neurologist, the FLAIR, where the white matter looks dark gray and the gray matter looks a little bit lighter. And this is an unaffected brain. And what I see most often are people who are getting diagnosed with multiple sclerosis, which is an inflammatory or acquired demyelinating disorder where the immune system targets oligodendrocytes and they die and you lose myelin and you get damage in the brain. And you get white matter lesions and they look white here and they occur on the part of the brain and it can be all over the place. And there are 1 million people currently in the US living with MS, and we have developed diagnostic criteria. We have over 20 FDA approved medications that target the immune system, but we have no medications that fix the brain once lesions are already formed. And that is what I've long been motivated to study in the lab. This is a bit in contrast with an inherited disorder, and I'm showing here actually the same MRI that you saw in Dr. Eichler's talk. This is just one example from the slide that he showed where the white matter here is now diffusely abnormal. And you can see it's affecting both sides. And that's and also in this case, frontally predominant. And so these are the clues that a more metabolic inherited disorder is going on because of the diffuse and confluent nature. And in this case, it's affecting a different cell type or microglia, and it's because the gene that's specifically expressed by microglia is altered. And so I started my lab at Penn Medicine in 2018, and basically working through the Comprehensive MS Center and the Center for Neuroinflammation and Experimental Therapeutics, where my own lab program is to study glial cells. And while I was a neuroimmunology fellow at Hopkins, I basically became increasingly frustrated with the number of cases that didn't quite meet criteria for MS or some of the more rare inflammatory acquired disorders. And so people like Ali Fatimi and Adeline Vanderver let me hang out with them and learn how do you do a neurogenetics workup on these adults. It was something I hadn't learned during residency. And so when I came to Penn, I was able to join not only the MS division, but the neurogenetics division to establish this undiagnosed white matter disorders clinic. And so in a study that was just published from our group, looking at all of our neurogenetics referrals between 2015 and 2020, so I started in the later end of that, white matter disorders comprise about 3% of them. And so the referral criteria for my clinic was basically if there are extensive white matter abnormalities that are atypical for more common diagnoses like MS, headache, or vascular disease, or there's a known family history. And this is a comprehensive care clinic where I evaluate everyone in collaboration with the genetic counselor. And typically, if there's no known family history, we do broad genetic testing, so a more agnostic approach where we send clinical whole exome, whole genome research or clinical. And now that Dr. Eichler mentioned that there's a sponsored leukodystrophy panel, we're able to send that as well. I also do workup for rare neuroinflammatory disorders and have in fact diagnosed these instead as a result of this approach. And so in this clinic, we're providing genetic counseling for both the newly diagnosed patient and family members. We're giving access to support groups such as what Heidi Edwards described for the Sisters Hope Foundation, which is an amazing resource. And we address treating symptoms because we can improve the quality of life, even if we don't have, in most cases, a disease-modifying treatment. So we can address seizures, tremor, anxiety, mood disorder, pain, spasticity, and sleep disorders. And we collaborate with other specialists like physiatry, physical and occupational therapy, urology, neuropsychology, palliative care, other medicine specialties, depending on the disorder, and social work. And I also collaborate with Adeline Vanderver over at the Children's Hospital with the studies that Dr. Eichler mentioned. So there's the Myelin Disorders Biorepository Project, and that CHOP is the lead site for that with over 2,000 enrolled families, and also for the Global Leukodystrophy Initiative that you just heard about. And so all patients who present to my clinic are offered enrollment into this effort. So we collect blood samples, imaging, and clinical data to de-identify with the goal to develop natural history studies and eventual clinical trial readiness, as Dr. Eichler mentioned, for specific disorders. And so just so you have an idea, this are my results basically from earlier this year, that of the 55 patients that I've assessed so far in this clinic, I have results from genetic testing in 42 of them. And I'm getting a genetic diagnosis in about a quarter of them. And the most common diagnoses I'm making are for X-linked adrenoleukodystrophy, and the second one is CSF1R related leukoencephalopathy. And so, because I've now seen it so many times, and it's frustrating to not have a disease-modifying treatment when this is very much in contrast with my MS clinic, I have sort of stumbled into realizing that I want to help develop these interventions. And so one of the most important things that we need to identify is how to get it, to find it early. So before symptoms or brain lesions develop, and we can do that. We have a very specific biomarker that we don't have in MS, which is genetic testing. However, unless you have a family history of it, we wouldn't know to test for that early on. So what kind of biomarkers do we need to be able to do that? And we can learn that by having natural history studies and by better understanding the pathogenesis of the disorder. We need to develop a treatment that prevents the formation of brain lesions. And you heard extensively about bone marrow transplant that seems to halt the formation of disease. But if we know more about the pathogenesis and targeting the function of the CSF1 receptor, then we can essentially more specifically target, I think. And we need to develop treatments that reverse damage in the brain. And we need this for all the disorders that affect the brain and spinal cord because they don't really exist yet. And so this sort of fits very nicely into the fact that that is something that is already the goal of my own research program. And so I'm going to now tell you a very basic version of what I imagine is going on in the brain based on the data that we have to put us all on the same page. And so on the left side, we have the blood vessel. So a blood vessel that innervates the brain with passing, you have the blood-brain barrier separating the blood vessel from the actual brain parenchyma, and inside the brain are all the neurons. I'm just showing you one that has, a cell body at one end, and it sends long projections to communicate with another neuron. And so within the blood vessel, you have, in addition to red blood cells, which I'm not showing here, white blood cells or immune cells, and they can be separated into innate versus adaptive types of immune cells. So innate cells, like macrophages, monocytes, and dendritic cells, I'm just showing you some examples here. These are the types of cells that respond to patterns, like invading pathogens, and sometimes from damaged cells. And they can gobble up those signals from the invading pathogens and then present some of those signals to the adaptive immune cells like B and T cells to help instruct them to develop a more specific immune response or a memory response. Within the brain are microglia, and microglia are basically the brain macrophages. And as you already heard, microglia, while very similar to those peripheral innate immune cells, actually come from the yolk sac during fetal development. And once they're in the brain, they self-renew, and they're separated by the blood-brain barrier from all of the other types of myeloid cells. Whereas in the blood, those circulating cells come from the bone marrow over the course of lifetime. But all of these cells express a CSF1 receptor. The other types of brain cells that are really important here and involved in the pathogenesis include oligodendrocyte lineage cells. So these are oligo precursor cells, and they're available throughout life. And they are the cells that are responsible for making those mature myelinating oligodendrocytes. But they actually also do many other jobs throughout life in addition to being a precursor cell. And we're only just learning about those functions now. There's also astrocytes, which I'm showing here as a puffy cloud because they basically fill in all the other details of the brain and interact with all of these cells and blood vessels and neurons and oligodendrocytes, microglia, and OPCs. And they coordinate the function of all these cells. And so how do brain lesions form in ALSP? So we know that we find fewer microglia within the brain when they express a mutation in CSF1R, that there's loss of oligodendrocytes and myelin, that the axons from the neurons are transected and damaged, and that astrocytes become reactive. So I made them pointy because reactive astrocytes, we're not even entirely sure whether that is good or bad or forming a scar, but they change their function. And what we don't really know actually is if the blood-brain barrier is affected and whether immune cells invade. And so Dr. Eichler showed that there can be a lot of macrophages found within the lesions. And so are those macrophages that are invading from the periphery, are they altered different types of brain resident macrophages that I haven't really shown you here? We don't actually know. The other thing that we don't know is the order that these things happen in. And we don't know whether what starts it in the first place. What was the first step that caused this to happen, and why does it happen in adults? So my goal to start to get at this is to develop a biomarker platform. So just going from the perspective of the peripheral blood, what markers happen in the cells that are either expressing the CSF1R mutation or not can be identified pre-symptomatically, symptomatically, and over the course of disease. And so to do that, I'm collaborating with Dr. Amit Bar-Or, who's the director of the Center for Neuroinflammation and Experimental Therapeutics, who I work with, who develops these peripheral immune cell platforms to better understand MS. And we can apply them to look at the cell specifically in this disorder. And my intention is to use the samples collected as part of the Myelin Disorders Biorepository project, but the best way to do this is with as many samples as possible. So if you're interested and you know people to refer, please send them our way. In addition, my own basic science lab is interested in studying the role of glial cells in acquired and inherited disorders of myelin. And that has very much been from the perspective of acquired disorders like MS. And I'm showing here a pink oligodendrocyte that we imaged in the living brain because it's pretty. But I just want to show you that we've developed this platform to monitor oligodendrocyte loss and replacement in the intact brain. And what I'm showing you here is a movie that you can access online at eLife. It's for free. From a paper that I did as part of my postdoc, where we looked at the same part of the brain over and over and over again when exposed to a toxin that kills oligodendrocytes, and then they're replaced by the endogenous oligo precursor cells. And so within this region, we can define what happens over the course of damage and repair, and also see that there's limitations. And in fact, microglia are probably playing an important role in that limitation. So the interaction between the two cells are incredibly important. And so we need to develop models to monitor damage and recovery in the intact brain in a primary microgliopathy like Alsp. Shown here are what these beautiful microglia cells look like just in an immunostained section, but they're actually very active cells. Shown here is the similar type of approach that I've used for oligodendrocytes, but what's called in vivo imaging of microglia. And shown here, and Dr. Eichler mentioned this, that microglia are the first responders. And in this video, there was a laser-induced lesion. I'm just going to start it again for you. You can see all the processes after this injury of the microglia are directed within minutes to hours to this area to clean it up and fix it. And so what happens when these microglia are expressing this mutation? Are they able to do these functions anymore? Is that what is impaired? Or are there other are they not able to do this at all in the first place? Or does an initial injury sort of step off a series of events that leads to the clinical phenotype of CSF1R related leukoencephalopathy? We don't know the relationship yet. And so I just wanted to show you, this is my new and growing lab. Hannah Liu here is my graduate student who's interested in studying microglia in this context. And so we're developing ideas and approaches to start to get at this from the very basic science level. I have down here my email address and our clinic number as well. But I just wanted to mention that as a summary of all of these things, that my goal, now that I've had 2.5 years of experience with this clinic, is that I intend to grow collaborative efforts with the Myelin Disorders Biorepository Project and the Global Leukodystrophy Initiative, multi-center and industry collaborations to develop natural history cohorts for this disorder so that we can develop a peripheral immune cell biomarker platform and identify this early as possible would be the goal, as well as develop clinical trial endpoints to test potential therapeutics. And then regarding the pathogenesis, we're just now developing the tools, and they're available to make better models to understand the pathogenesis in ways that allow us to understand cell-cell relationships in the context of what could be going on, so that we can identify effective and targeted therapeutic interventions. And so with that, I would just like to thank, I have to work with all of these people who helped me establish this clinic and research program, as well as people that trained me in doing this approach as well, and the patients and families who are living with it. Thank you.
Well, thank you. Those were excellent presentations. If anyone has any questions, you can type them in the chat or use the raise hand option located under the reactions button at the bottom of the screen. We do have a couple minutes left if anybody has any questions. Wow, I guess you guys were really thorough. Oh, did you want to put your mic on?
Go ahead, Elliot.
Do you hear me? Can you hear me now?
Yes.
I have a daughter who's 48 years old right now, and she was diagnosed 3 years ago with the CSF1R. She was initially diagnosed as having frontotemporal dementia. We took her to a tertiary care facility where she was correctly diagnosed, but we were told that there was no treatment for the disease. I was fortunate enough, being a physician, to contact Dr. Eichler, who saw her and recommended a transplant. Unfortunately, her insurance declined to have it done at Mass General, and we enrolled her in the clinical trial at the University of Minnesota. She just had her 2-year anniversary of her transplant. From all perspectives, the transplant has been successful in stopping the progression of the disease, at least by MRI. She's physically fully active, walking 2 miles a day. The negative was that post-transplant, she suffered major cognitive impairment, which seemed to follow her chemotherapy. And also from my perspective as a physician, when she received large amounts of granulocyte stimulating factor for severe leukopenia, her cognitive function really deteriorated. The cognitive function has improved a little, but not a lot. A lot of what she is doing now is relearning activities. But I just want to make you aware that she was the first one to undergo the transplant at the University of Minnesota. And thanks to Dr. Eichler, we pursued that, because I feel very confident that if we wouldn't have pursued that, that she would have died by now. So I just wanted to pass that on. I think there needs to be a lot of investigation into why her cognitive function deteriorated post-transplant, from both the chemotherapy, the chemo brain, and also from the perspective of the granulocyte stimulating factor infusions. Just thought I'd make those comments.
Thanks, Elliot. I think very, very important observation. And I think there's much more crosstalk occurring between the bone marrow and blood and brain, and we understand. And so what you're mentioning here, and any observation that you're making that is consistent and really a lived experience of patients, goes a long way. And so I would just encourage you to keep reminding us of this. There are adjacent fields, and Dr. Orthman Murphy is bringing so much knowledge and information from the field of MS and neuroinflammation into this. But we also need to lean heavily on our transplant colleagues and knowledge around hematology to understand what is happening here. Because I think you're right, there can be effects through GCSF and other chemotherapy that we have not fully understood yet. But I think it's also really an opportunity for improvement. If there is resilience, we will come up with better neuroprotective agents, and we might be able to help carry in the future with this. So
Yeah.
lovely to see you again.
Thank you. Thank you for all your help.
Thank you for sharing that. It's really, like Dr. Eichler said, really important insight into understanding that, while there's hope for having the possibility of a bone marrow transplant, it's not perfect, and we don't really understand the consequences of that either. And there's preclinical studies in animal models looking at immunosuppressive therapies like methotrexate that have shown that it completely disrupts and causes loss of those oligodendrocyte lineage cells like the OPCs. And in those studies, it suggested that that could be somewhat the basis of chemo brain. So perhaps, these treatments are also interfering with the ability of the brain to repair itself, but we don't know in this context without doing the studies and having the right markers to really understand that. And I think collaborative efforts like this for a rare disorder where we can combine our efforts and know that these are the things that we need to look for are really important.
And just to reiterate what Dr. Eichler said, the bone marrow transplant was brutal. She spent 4 months in the hospital in Minneapolis. She had graft versus host. She had a number of infections. She had persistent leukopenia for 1.5 years. And finally, after 2 years now, she has a normal weight count. So it was a very, very, I used to have a full head of hair, by the way, but it was a brutal course. But I'm glad we did it.
And I think it speaks to the strength of your family, to having pulled through and having ensured that she's done so well. We often forget that this disease does not only affect a single person body, but the entire community and family. And likewise, it needs entire family to pull through this. And the kind of support you're giving to her is what I've seen as a big determinant of outcome when going through a transplant. So not to underestimate that.
Well, my other important point is that if I was not a physician, I was basically told her to take her home and basically watch her die. I would have never pursued further treatment. I would have never found your article. I would have never found the clinical trial. So clearly, if you're a layperson, it's very difficult for this to be diagnosed and for potential treatments to be offered.
Heidi, can you speak a little bit to what else you think we're missing and what kinds of things we physicians, scientists should think about?
Well, and I was going to thank Elliot for making that comment. In 2009, when my mom became symptomatic, I decided I would not let her. At that time, they told us just to go home. There's nothing we can do. And we watched her just suffer with this disease. And I thought, I can't let other families do this too, go through this. I spent every day researching and trying to find articles and more information on this disease. And I was getting nowhere. And finally in 2012, when the CSF1R mutation was found, it was such a breakthrough, but we still have so much to do. Here I'm going to lose both of my sisters in their mid-40s to this disease because they waited too long, and I was an advocate to be tested early. I, in 2014, had genetic testing done. I know I am negative for the mutation, but my sisters, they did not want to push. They did not want to know what was going to be their fate. And that was a downfall for them. By the time we finally talked them into going to the hospital, meeting with the doctors, getting tested, they had progressed so rapidly that Heather, she was at that cliff, they said. And when she started the transplant, she had the chemo, the transplant, she went over that cliff with progression and couldn't nothing could be recovered for her. It was too late. Holly was at that point, we felt, and Holly knew she didn't feel that she was going to be able to survive the transplant either. So through Sisters Hope, what I envision is making sure that Elliot, like you and your family and my family, others do not have to dig deep and try to figure it out on their own. We have the resources right at their fingertips. So that if you are not a physician like you are, Elliot, they can easily understand this disease and easily find the resources that they need to make an educated decision. And thanks to Dr. Eichler and Dr. Orthman, they will help bridge these gaps and the communication for patients in the future.
Yeah, I think we're going to see so much more early diagnosis in the future, and it's going to be a big game changer. So I think also recognizing that there's new technology now becoming available, and many partners in drug development that can also help. I think we're going to see a paradigm shift.
Okay, well, if there are no further questions, I just want to thank our speakers and our sponsors and our audience for making the virtual family conference this year a possibility. We hope to see you in person next year. Thank you to everybody.
Take care.
Thank you.
Take care.
Thank you.